If you pay attention to clinical trials, you've probably heard the term statistically significant. For example, with a p-value of 0.05, the results were statistically significant. And you might have wondered, what does that really mean? The whole point of running a clinical trial is to demonstrate that a new drug or procedure or medical device is better than placebo or the standard of care. And in order to do that, you have to gather a whole bunch of data. But before you can make any compelling claims of success, you have to dip into the magical world of statistics. So any statistical test is an effort to prove or disprove what's called a null hypothesis. Using the example of a clinical trial on a new drug, our null hypothesis would be that the drug doesn't work. And so our goal is to prove that hypothesis wrong. This is where the humble p-value comes in. It's short for probability value, and it represents how likely your clinical trial result would be if the null hypothesis were true. Said another way, the lower the p-value, the more confident you can be that the null hypothesis is false, i.e. that your drug works. So when you hear that phrase, the p-value was less than 0.05, what that means is the researchers did a bunch of math and based on all the data concluded that there's a less than 5% chance that the result in question was due to random chance, hence statistically significant. But statistics is not gospel, and just because a given drug meets that 0.05 threshold doesn't mean it actually makes a difference in the lives of patients. For example, if you run a big enough trial, you can actually demonstrate statistical significance on what turned out to be a pretty small absolute benefit in terms of what patients actually experienced versus placebo. Then there's the pernicious practice known as p-hacking, which is when researchers pick and choose from the data in order to present a more attractive conclusion and reach that magical 0.05 number. So while it's by no means a perfect tool, the p-value is still invaluable to protect the process of science from the effects of random chance. Without it, or at least something like it, it'd be very difficult to know whether a given trial was a slam dunk or just a lucky roll of the dice.